Good afternoon, everyone. I want to thank you for being here. I'm a PhD candidate at the University of Cincinnati, but I happened to find a few plants up in the mountains that needed some researching, so I'm going to tell you a little story about them in terms of where I'm at so far with my thesis work. So I'm going to be really brief on the background information just because I think a lot of us are very familiar with habitat fragmentation, but I've got a lot of new results that are really hot off the press that I would prefer to share. But briefly, habitat fragmentation is a very bad thing, just thinking about in terms of biodiversity. Maybe not so much, it depends on who you ask, but for actual species and individual populations, it can actually be a terrible thing in terms of habitat loss as well as increased isolation. And we can see this in the form of short-term effects in genetics based on decreased heterozygosity, which can lead to reduced fitness. And then also, if you think on a longer time scale, we can also see higher susceptibility to environmental changes. And call it my naivety from coming from the Midwest, but I usually think of habitat fragmentation from they're putting in roads, dropping in suburbs, whatever else it might be, where we have this really powerful anthropogenic effect on fragmentation. But there is this little thing called mountains, where they actually, a lot of these alpine meadows see habitat fragmentation much more frequently and on much longer scales. So this is Jumping Pound Ridge in Alberta, Canada. This is 16 years after the Camp Fire, which was this massive wildfire that they had back in the 30s, where it actually had a scorched earth where you expose bedrock because it burned so hot. But we can see from the 50s up to just a decade ago that you can see how the forest has gradually sort of encroached back in, presumably isolating these alpine populations. And just to give you a sense of what these areas look like today, well, last year, you have these really broad open meadows, and then you can also see the forest sort of encroaching coming up off of those, from those lower elevations as well. And so what I wanted to do was look at genetic variation present along this particular ridge line of Jumping Pound Ridge and see how much or little there is within this one particular species that I'll tell you a little more about. And then I also wanted to look at connectivity among what we're assuming are actually isolated populations as well. So briefly, I would focus on the lanceleaf stonecrop, Sedum lanceolatum. It's a small succulent that'll fit in the palm of your hand, and they generally will grow for 1 or 2 years in bolts, and then after they set seed, they'll actually, that rosette will die off. But still have a lot of these usually vegetatively sort of grown off of the same rosettes, maintaining that same individual. They are pollinated by just about everything and their mother. And they also, the seed morphology, it looks like they are carried by something with flow, namely being water or by wind. And they are broadly distributed, which I'll show you here in just a moment. And one fun little wrinkle to it is that they are usually diploid, but they also exhibit varying degrees of ploidy out in the wild as well. So it is broadly distributed from New Mexico all the way up into Alaska. And some work back in 2005 focused from Colorado up into Montana, and they found these huge amounts of genetic variation using chloroplast markers across that range, looking at samples within national parks. And so that got me thinking too, well, how much do we see at these smaller scales? And so I'm focusing on 17 meadows that run along that Jumping Pound Ridge. We saw this map a little bit earlier. This one, I just have our meadows actually sort of called out with the polygons. And a lot of the work done at this area, which is helpful for me, is on the Rocky Mountain Apollo butterfly, to which this plant is their obligate caterpillar food source. And so I'm calling the meadows just as they do in their studies, but one nice thing about this, as I said, is that we have DNA samples that kind of go back through time as well. So to start addressing some of these questions of variation, I use a traditional library enrichment approach, just using biotinolite probes to try and identify repetitive fragments. And then sort of winnowing down, I ended up with 6 polymorphic loci. And to actually run through amplification of these loci, I wanted to incorporate touchdown because it's a little bit faster and incorporates, or it reduces non-specific binding. Cache is limited, so I wanted to use multiplexing, so I can throw a whole bunch of different microsets together into one mix. And then we also use dual labeling so that you can use, it's a lot cheaper than having to buy probed individual specific markers. The one downside to this approach, touchdown is good for when you have high annealing temperatures in some primers and low annealing temperatures in others. The downside is that you end up with all the primers at this much higher annealing temperature are going to crowd out the loci that they amplify early on, to when you get to the lower ones, you have much less opportunity for amplification. So I did something where I actually kind of tweaked some of the concentrations to it, and that actually helped, I'll tell you a little bit later, this is actually, I think, a really powerful approach if you want to chat more about it later. So for what I'm going to show you today, last summer, which is why I didn't make it to Botany, I collected over 450 samples across these different meadows. These meadows are extremely large. That is the width of a road, just to give you a sense of spatial scale. And for here, I'm going to show you 352 individuals that are actually in the analysis. So we went out, grabbed leaf tissue, left them desiccated, and then we used CTAB extractions once we got them back to Cincinnati in the lab. Then I used Popper and EcoDist for some of the analyses. So looking at genetic variation, they're not that terribly different. It does seem like there is some patterning to it, but I think it's just because I squint too much at it. And additionally, when we look at Hendrix, GST, and GOST, we can see they're negative, which is effectively 0. So there is no differentiation among genotypes, and likewise among alleles within these different populations. So that was a little bit of a bummer, but I kept on hard charging at it. So I took centroids of sampling within each of these populations, and then we have pairwise distances within each of them. And I decided to run, or I used a Mantel test to see if there's any sort of relationship between geographic distance and genetic distance. And there's not. You can see these really clear lines sort of suggest that there is a lot of within population variation rather than among population variation. But I didn't stop there. I was also curious about the role of forest in terms of being a barrier to either seed dispersal or pollinators. So I took those same lines and I was able to pull out where you do not have forest, so we can actually look at the effective forest distance that you have between each of these different populations. And because geographic distance is sort of inherently within forest distance, I ended up using a partial Mantel to control for that geographic distance. And with that, we still get this random smattering. Oddly enough, it does come up significant, but it is ever so slightly negative, which I think again shows that there is just a lot of gene flow among these different populations along these meadows. And so I wanted to continue with this, and so I used a Bayesian analysis. And the way that this works is we're looking to see if there is any sort of directionality in terms of genetic distance, geographic distance, and that actual angle that you see pairwise between each of the different things that you're comparing. And so to make this a little bit easier to interpret, we'll just imagine our field and then a compass on it. And so we can see these solid dots kind of tell us where there are significant correlations, and then these X's are where they're non-significant. And so below we have a negative correlation, sort of going from that like southwest to northeast directionality. So there is no correlation there. But then we do see from this other side that there is positive correlation, sort of going from this northwest to southeast directionality instead. And if you kind of look at how things shape out across this landscape, we can see that it does seem like there might be something to do with like nuances of the actual landscape that give us some sense of continuity among these populations than strictly speaking just looking at them as a whole. I know what you're thinking. Why would you expect to find differences at such a small scale? The study that was done before, going from Montana all the way down to Colorado, they used chloroplast markers, which are much more conserved relative to nuclear markers. But they also did it over a tremendous scale, so I wanted to kind of winnow it down to a much smaller size. The other piece is that there's only 6 loci. I'll get to that in a minute. So we do have some sampling from 2017. The Jumping Pound Ridge, that's where we were at just a minute ago, along with Cox Hill, which was that one that was in the center of east to west. We have these 2 other ridges that are northwest and also southeast, Lusk Ridge and Powder Face. And we have some sampling from those, which give us up to a 20 kilometers distance see them within this system. And so looking at the PCA, we still see that there is a lot of similarity among them, but you do see that they do somewhat parse out. But again, GST and D both say that there's a lot of fixation and very little differentiation among these. However, when we look at a Mantel test, we do see that it is a little bit more normal in terms of there is actually isolation by distance as well. So back to the only 6 loci thing. It is not ideal, I admit. But when I figured out, playing around these concentrations in PCR, I was actually able to recover an additional 9 loci. And I do not have the data here to show you at this moment, but it shows me very similar results as to what I've presented here in terms of negative fixation and differentiation, and then also very limited sort of relationship And with that, I feel pretty confident in the 6th, but don't worry, I'm going to go back through and do the rest of it. So thinking about this, there's very little variation among populations at this small, 20-some-odd kilometers scale. But we find the species across, the Rocky Mountains as a whole. And it's interesting to think because some of the variation that was found previously along the much larger scales, it's thought of that as glacial and interglacial cycling. So we're forcing populations down during glacial periods and then forcing them back up due to trees during interglacial periods like we're in now. But it seems that there is some more local, regional variation that is maintained, which is, I think, what I'm sort of detecting here. But that's super speculative at the moment. So some of the next things is, I mentioned how there are some sort of patterns within the landscape. So my next step is to sort of look at these in individual demes to see if there might be some substructuring that we can actually detect. I am going to throw in those additional loci that I mentioned as well. And the last thing is another piece of my thesis work is looking at trying to describe population growth within the species using climate meteorology. And since we have samples that go back to 2009, I want to try to incorporate some of that genetic information to see if there are any sort of continuities between changes in population sizes and then also with the genetic variation. Because my one thinking is that when you have a population crash is if that's going to homogenize genetic variation among all these different populations as well. But we need to tie that with population sizes as well as some of these other abiotic factors. So with that, I want to thank my advisors and co-authors, Theresa Culley and Steve Motter, my committee members, folks that gave me money, and my kick-ass undergrad students that helped me out in the lab. So, thank you, and let me know if you have questions or comments.